Good morning, everyone. My name is Samantha Riles, Communications Officer from Sugar Research Australia, and I'd like to welcome you to today's webinar. Please note that today's meeting is being recorded and will be available on our website. At any time that you are not speaking, can I please ask you to remain on mute to keep the background noise to a minimum? If you're having any technology issues, please send a message in the chat box, which is located on the menu bar of the screen, and I will be acting as a co-host to ensure all runs smoothly. We would like today to be an open discussion, so if you have any questions throughout the presentations, please turn your microphones on and ask our presenters. We will also have questions at the end of each presentation, and we encourage you to take an active part. You can also type questions into the chat box throughout the presentation, and Seb and I will moderate these at the end. I'd now like to hand over to SRA's New South Wales Regional Coordinator, Sebastian Garcia, to introduce our speakers.
Thanks, Sam. It would probably have been easier to say my name as per Rick, Serge Gainsbourg. Thank you all for joining in. As you know, we had to cancel this year's ACT seminar, but we thought we could still hear from some of the work that was going to be presented anyway through this type of media. Pointless to say that nutrient and more specifically nitrogen management is a critical aspect of sugarcane farming. In New South Wales, our soil types, our weather and farming system tend to greatly influence the way nitrogen is used by our crop. Weather and how we may lose some and what may remain, for example, after a crop of soybeans. Ultimately, the way we manage that nitrogen affects farm productivity, profitability and sustainability. So today we have two of our local researchers. We've got Dr. Lukas Van Zwieten, which is a principal research scientist at New South Wales DPI, and Terry Rose, Professor of Sustainable Cropping System at Southern Cross University. And they will both talk about their latest work. Lukas will be talking about his research as part of the More Profit for Nitrogen project, which looked at soil existing nitrogen sources in New South Wales sugarcane soils and how to measure these. Lukas will also present the results of his trial comparing polymer coated and standard urea fertilizers. Terry, in previous research, found that there was substantial amount of nitrogen produced by soybean that was actually lost before planting the cane. And that planting winter cover crop may be a way to both capture the nitrogen and improve soil health. So Terry will be giving an update on his last winter's trial. So Lukas, overtake it.
Thank you very much, Seb, and thank you as well, Sam, for the introduction. As Seb mentioned, my presentation today will be some of the practical work we've been doing around nitrogen in the sugarcane farming systems, and also a snapshot of the data and the summary of results from our field trials on polymer-coated urea. This work has been supported through the More Profit for Nitrogen program, which was run by the Rural R&D for Profit program, an initiative of the federal government. SRA, Sunshine Sugar and Southern Cross Uni are also acknowledged here. I've got lots of project participants that have had input into the project. You can see them here, and in particular, again, I'd like to thank the farmers, Jeff Pye, Alan Munro, Rob Quirk and Wayne Rogers for enabling our field trials to go ahead. Okay, so the presentation today is just going to review what we found with regards to the nitrogen stocks in the New South Wales sugarcane farming soils. And what we're hoping is that down the track we might be able to incorporate some of this newer information into a refinement of the 6 easy steps That is still being worked on. One of the aims of the polymer-coated urea is that we've better synchronise the nitrogen supply with the crop demand. And I'll show some of the data there on the nitrogen release from the polymer-coated urea. And also, probably more importantly, again, we've got a new technology for the rapid prediction of the soil supply of nitrogen, and that's called the potentially mineralizable N. And I'll just go through that in a few moments and explain that a little bit more. So the main sources of plant available N or crop available N are obviously the fertiliser that you apply, i.e. urea or whatever other form of fertiliser is applied, and that could include organic amendments as well. You've got an existing stock of nitrogen in your soil, mainly as ammonium and nitrate, and many farmers or agronomists will often measure, the 0 to 20 or 0 to 30 centimetre soil layer, but what we're doing in that particular process is ignoring the deeper soil stocks of nitrogen, which I think sugarcane plants can actually access. So in our assessment of mineral N stocks in New South Wales, we had looked down to a metre, and some of the results were quite surprising. The other source of plant available N is through the breakdown of plant litter, i.e. legumes that Seb mentioned as a part of Terry's work, and also the sugarcane trash. And as this organic material breaks down, it can release nitrogen into soil. And also you have the breakdown of soil organic carbon. So that sort of fixed carbon that's in soil, when that breaks down, that too releases nitrogen that can be taken up by the plants. And importantly, that sort of combination, that sum of 0.3 above and 0.4 above, which is a breakdown of plant litter and the breakdown of organic carbon in soil, is what's known as a potentially mineralizable N. So that's a nitrogen again that comes. That's the nitrogen that comes from the breakdown of organic materials in soil. We also need to understand soil carbon to understand that potentially mineralizable nitrogen. So legume biomass has got a carbon to nitrogen ratio of around about 12 to 40 to 1. So that means for every carbon, or say for every nitrogen, which is a 1, there will be 12 to 40 carbons. If you have a look at cane trash, it's got a much higher carbon to nitrogen ratio, 70 to 1. And native soil organic carbon has got a ratio of about 12 to 1, which is very similar to some of the legume biomass. So what that means is that the legumes have got a lot more nitrogen on a per weight basis to supply in potentially mineralizable nitrogen than, for example, cane trash does, or even other woody materials. So we had a look, this is back in 16-17, had a look at the nitrogen stocks from 27 cane paddocks in New South Wales, and we broke it up roughly into the Tweed, Richmond and Clarence catchments. And apart from doing all the other soil tests along with these, the main thing that we looked at was the mineral N, which is ammonium and nitrate, as well as this potentially mineralizable nitrogen in the 0 to 20 and 20 to 40 centimetre layer. So that PMN test, even though it is accounted for in the 6 easy steps, it's not something that is routinely measured because it actually takes a long time to measure it, and at the moment it's quite an expensive test to do, but it can actually provide a fair amount of nitrogen into the soil. Now, this is not the prettiest graph in the world to try and understand, but I'll just go through it over the next couple of minutes because I think it's quite important. So here we have along the x-axis the total nitrogen per hectare. So starting from 0 all the way up to 350 kilos per hectare. This is just the number of sites that we had a look at. So the red sites represent the Clarence catchment, the green sites represent the Richmond catchment, and the blue sites represent the Tweed catchment. And what this is showing is that the majority of sites have got ammonium in the soil at around about 25 to 50 kilograms per hectare of available ammonium. And the same with nitrate. Most of the sites actually had no nitrate whatsoever, or very little residual nitrate. However, when we add the 14-day potentially mineralizable nitrogen, so this test is a test that takes 14 days to analyze, we actually find that some of these sites have got up to 200 kilos of nitrogen as PMN. So that's the organic material as it breaks down will release 200 units of nitrogen into soil. So we add all of these together, and basically what it shows us is that some of these sites have got roughly 1/3 to 1/2 the sites that we analysed have got between 200 to 350 units of nitrogen from existing stocks of mineral N and potentially mineralisable nitrogen available in the soil already. There are clearly some sites which are in a strong deficit. So down the track, can we use this particular information on existing nitrogen stocks and PMN to better estimate how much nitrogen the crop will require as a fertiliser amendment to supplement the soil supply. Again, the big difference here has been the PMN, this potentially mineralisable nitrogen component. So these tests take 14 days. We also do what's known as a 56 and a 300-day potentially mineralisable nitrogen test. The problem with that is by the time you get the data back on how much nitrogen is mineralised in the soil, it's too late. Basically, you're 14 days or 56 days down the track, and it's too late then to adjust your nitrogen dose. So what we needed to have is a way to estimate what this potentially mineralisable nitrogen will be in a rapid test. And this is just some PMN data from the Clarence catchment, 8 sites that we had a look at there. And you can see in 7 days, most of these sites released between 0 and 20 kilos per hectare of nitrogen. But over 56 days in the top 20 centimetres, one site released 60 kilos, another site released 20 kilos. And knowing that difference, depending on the history of the paddock and the soil type and exactly where you are, would be a really useful thing to be able to measure. Likewise, sorry, Seb?
Sorry to cut you. We've got a question here from
yes.
Andrew Dougal, which may be relevant at this stage. He's asking if all of these measurements are total nitrogen.
So this PMN is basically development of nitrate.
No, Lucas, on the previous slide, there was 4 measurements. Do all 4 of those equal do all 4 of those equal the total N?
Correct, yes. So the ammonium plus a nitrate plus a mineralisable N equals a plant available N per hectare.
Total N.
Total N, yeah. Well, no, not total N, total plant available N, because total N is quite different to plant available N. So this is a sum of mineral N in soil.
Okay, thanks, Lucas.
Yeah, sorry about that. I should have explained that more clearly. The Richmond catchment, the soils tend to have a greater amount of PMN, possibly because they've got a slightly higher organic carbon content in general. And some of these sites produce nearly 80 kilos per hectare of mineralisable nitrogen. And we look at the Tweed Valley. So this is some new data that we've just generated. Again, some of these sites have got a lot of mineralisable nitrogen in the surface. Some a lot less, to 30 odd kilos, some up around 80 kilos. So, what we have here is a good idea of the range of PMN in soil. And as I mentioned, the problem with doing something like a 56 or a 300-day test of how much nitrogen will mineralise in your soil across the crop season is a test that takes a long time to do. And as I mentioned, by the time that you actually get the data back, you've missed the boat there on adjusting your nitrogen rates. So what we've used a test called a mid-infrared spectrometer, or MIR. So it's not actually a test, it's a prediction. And this particular machine basically looks at reflectance of an infrared light source that's put onto the soil, and it can predict if you've got a good calibration, if it's done enough tests of a particular soil, you can then start predicting certain characteristics. And for example, CSIRO have come up with a prediction for soil carbon. We've used this particular type of machine now for predicting what's known as this potentially mineralisable nitrogen. And what we have is essentially a very strong calibration curve that should be able to predict it for the New South Wales soils that we've got a lot of data on and we've got a strong calibration on. It's 14, 56 and 300-day mineralisable nitrogen component. And essentially this is a $30 test which can be done overnight. So, you know, we've got all of the chemistry, that wet chemistry data in the background that's been used to develop this calibration. And now we can basically go to a simple, cheap overnight prediction, and it will be maybe plus or minus 20% confidence around what the actual mineralizable nitrogen will be at either 14 or 56 or 300 days. Okay, now I'm going to just step across to some of the work we've done with these polymer-coated ureas, and just a little bit of a theory on why technically these polymer-coated ureas should work. So this is a fairly standard graph on the left-hand side here, and we're looking at total plant uptake, and this is just time on the x-axis. So you can see here, as you know, soon after the plant's planted, or in this case maybe harvested and you're retuning, the actual requirement for nitrogen isn't all that great. The majority of nitrogen is required during this rapid growth phase. So in the New South Wales sugarcane systems, that will be probably sometime between December and April. And then as the crop matures, again, it requires much less nitrogen. The actual N accumulation is a lot lower at that point. The problem is in a lot of the farming systems, and sugarcane is not unique here, the nitrogen is often applied up front or near sowing when the nitrogen actually isn't really needed by the crop. And what that does is that means that you've got a period, in this case with the sugarcane it might be somewhere between 1 month to 3 months, where that nitrogen that you've applied as urea is exposed to loss pathways. And I don't have too much time today to go into those, but the loss pathways could include leaching. If you've got heavy rainfall, those loss pathways could include denitrification, so loss of nitrous oxide or dinitrogen. They're probably the two key loss pathways in sugarcane farming systems. And the aim of the polymer-coated urea, as for example also products like NTEC, is to try and keep that nitrogen in soil longer, so that nitrogen supply is being released when you've got that demand of nitrogen. So we've got this sort of a couple of different polymer-coated ureas that we tested, the orange one and the blue one. The orange one was a 90-day release polymer, and the blue one was a 270-day release polymer. We didn't actually trust the manufacturers in telling us exactly what the release pattern is, so we analysed that for ourselves. And we use these litter bags in the sugarcane row, sort of exposed to the normal field conditions, to assess how much or how quickly that nitrogen moves out of that polymer coat and into the soil, which then becomes plant available. So this is at Korakai between November 2018 and December 2019. And we had a look at the polymer coat 90, which is a blue curve, and then the red curve is a 270-day polymer coat. And what we can see here is that they basically do what the manufacturer has suggested. So the 90-day polymer released most of its, probably about 80% of its nitrogen within 90 days, and about half the nitrogen was released within 30 days. And if we have a look at the polymer coat 270, about half of the nitrogen was released within 91 days. That means that sort of 3 months after the polymer is used, you've still got about half of the nitrogen which then moves out of the polymer and into the soil. So this is good. So theoretically what we're doing now is supplying the nitrogen from the fertiliser into the soil when the crop needs it most. And a very, very similar data set for Woodford Island. It's almost identical. We had 30 days for the polymer 90 and 90 days for the 270. We do need to note though that both of those sites had particularly warm and particularly dry conditions. So that release pattern may change, with more moisture in the soil, and we don't have data for that. Okay, so these are the field trials, and you can see here we've got a blend of the 270 and 90 and the 90-day polymer, and it's just applied by the conventional stool splitter, and we applied it at different rates in the soil. So everything from 0 up to 400 units of nitrogen, and we had I think 7 rates along the way. And we did this across 4 field sites. So the first field site was up at Stotts Creek, and this is the data that we had. So we're comparing different rates of nitrogen between either the polymer-coated urea, the PCU, or standard urea. So no nitrogen gave us a yield, and this yield was measured through the commercial harvester with a 5 kilo weigh bin. Was averaging out around about 80 to 90 kilos of dry mass per hectare. And we had an increase in yield up to around about 200 kilos of fertiliser, where the yield then plateaued off. Unfortunately, we could see absolutely no difference between the polymer-coated urea and the standard urea. So our hypothesis here was shot to pieces. Again, we thought that the polymer coat, because it would release nitrogen when the plant needed it the most, would give an improvement in yield, but that wasn't to be seen. So we went to Pimlico field site, which was set up again in December 2016, and measured and yield was assessed through as 2-year cane. No nitrogen, so we had a stronger response curve here. So no nitrogen again gave us around about 90 kilos for a 2-year cane, and that yield basically increased up to about 150 tonnes of cane at 400 units of nitrogen. Again, we saw absolutely no separation between the polymer coat, which are the orange ones, and the standard urea. So again, from the Pimlico field site, we couldn't recommend the use of polymer coat at that particular field site. Our third site was at Woodford Island. We're starting to pray for rain at this stage because that both of those previous sites have been quite dry, and we weren't experiencing those nitrogen loss pathways that New South Wales, I guess, is known for in that early summer period. And this particular site had a very, very flat response curve. So while the yield was still increasing up to 400 units of nitrogen, again, there was absolutely no separation between the polymer coat and the standard urea, and there was a fair bit of variability at that site as well. We had a look at leaf end content at the Woodford Island site, and what we certainly noticed here was that the higher doses of nitrogen, be it as polymer coat or a standard urea, certainly resulted in higher leaf nitrogen content. We could see that quite clearly. So the control had much lower leaf nitrogen content, and as our nitrogen doses increased, so did our leaf end content. This didn't necessarily translate, though, to improvements in yield. And what we also did at that site, we did a very intensive UAV, unmanned aerial vehicle assessment using multispectral imaging for the site, and running a whole heap of different indices, like you might have heard of NDVI, but the one that we have settled on is known as a green NDVI, the GNDVI. And what we found is we basically did the third leaf, SRA third leaf end method from soon after returning right through to harvest. And at most other times, we couldn't really find a very good correlation. But at this 160-day point, which also corresponds to some of the work that Mike Bell and I think Tomo have done up in Queensland, where the 160-day leaf analysis correlates to an extent to yield. We also found that the GNDVI correlates very nicely to leaf end content at 160 days. So that is potentially an assessment that we can use in the future to assess field, the performance of field trials. And you can see here, these lighter plots were the ones that would have had lower doses of nitrogen applied. And just quickly to our fourth field site, again, this site was at Coraki. Again, we had an extraordinarily dry start to the season. I think it was a 1 in 100 year dry start. And we've had a very, very flat response curve. And again, no separation between the PCU, or the polymer-coated urea, and the standard urea. So the key result, generally, we've had fairly flat nitrogen responses. However, it is, of note, and we've discussed this with Rick, that even at the higher doses of nitrogen, you still are getting some response to yield. At 4 field sites, so it's a pretty robust data set now. At 4 field sites, we've seen no difference between the polymer-coated urea or the standard urea. So, it really puts into question here in these dry years, in particular where you've got a dry start to the year, it puts into question the value of the enhanced efficiency nitrogen fertiliser. Again, I really need to stress here that all 4 sites had well below average rainfall, both for the growing season, but in particular at the start of the growing season where nitrogen loss pathways are prevalent. I've already mentioned that the GNDVI shows promise at 160 days after return emergence for estimating leaf end content, and the sugarcane yield was probably more limited by water constraints than nitrogen constraints at all 4 field sites. Okay, so some outcomes for the industry. As I mentioned, the polymer-coated urea did not influence yield, again because it had low nitrogen loss pathways. I think we can use mid-infrared spectroscopy as a useful prediction for mineralizable nitrogen. So at the start of the season, perhaps a soil test for ammonium and nitrate, and then also PMN could potentially refine some of the 6 easy steps. And I still need to work with Bernard Schroeder and some of the others like Rick Beattie on that to see how we could potentially use that information in some of those models. And as I mentioned, the multispectral imaging is used for predicting 160-day leaf nitrogen content. My recommendation again is just use standard urea unless you're going to have a very, very wet start to the season where some of these enhanced efficiency nitrogen fertilisers are more likely to work. And I think, and I've certainly mentioned this to SRA, that what we need is improved climate modelling and use some of those models then to better understand what years these enhanced efficiency fertilisers are likely to work in. And as I mentioned before, deep soil nitrogen and 56 or 300-day PMN can be considered in making some nitrogen recommendations for the growers in New South Wales. I think that's it, Seb. So thanks everyone for your time. I would have much preferred to give this as a face-to-face where we could discuss these results in more detail, but hopefully when we come out of the lockdown we can revisit some of this information.
Thank you, Lucas. I cannot see any questions that have been asked during the talk. Maybe just one quick comment. It seems to me like the polymer-coated urea the jury is still out, but one question from your experience or interaction with other researchers or projects, has there been any significant or positive results elsewhere in the industry?
So, yeah, great question, Seb. So the polymer-coated urea actually does what it's claimed to do. We were very, very confident that, and we've got strong data in the field showing that the release patterns of nitrogen from the polymer coat did exactly what the manufacturers said they were going to do. But where we had that dry start to the season, unfortunately, the polymer coat actually wasn't needed in those particular 4 seasons. There have been some great examples, in particular in Victoria, where you might have extended periods of wet and denitrification of nitrogen, so losing it as a volatile form of nitrogen happens a lot. They've had some quite a bit of success with enhanced efficiency fertilisers there. But yeah, in these particular systems, unfortunately, the seasons were particularly dry when these trials were run, and we didn't see that benefit of the PCU.
Okay. Well, thanks very much, Lucas. And if anyone has got any questions later on after the webinar is finished, just email them to me or give us a call, or if you've got Lucas's details, contact So now we'll go through Terry's presentation. Go ahead, Terry.
Thanks, Seb. You can see my screen, can you?
Yeah, all good.
Very good. I'll just make one quick comment about the Lukas's results first. Another thing I think it's worth noting, even with a wet season, when we're banding urea at, you know, a couple of 100 kilos per hectare, there's work from Queensland and it's, you know, been known for quite some time. When you band urea, you actually get some protection anyway. It doesn't form nitrate in those bands. You sort of get a toxic effect that'll hold it. And so even with modelling, we kind of, it'd be very nice to know how long do you get protection with just a banded urea. And then you can start to run modelling over the top with rainfall, because even if you get heavy rainfall a couple of days after application, you know, we're pretty confident that the urea hasn't become nitrate by then. Anyway, that's just food for thought and something we can discuss later on. So I'll switch to talking about cover cropping, winter cover crops in New South Wales cane systems. Firstly, thanks to a few of the guys who took a lot of the measurements for me, Anders, Lukas and Shamsul, and also thanks to Marty Walsh and Jeff Pye for hosting some trials. Sorry, I'm just trying to move my screen. So the reason, one of the main reasons we started this cover crop work is we were asked by some farmers and advisors whether or not nitrogen in in soybean residues, is all of that available come cane planting time? So we've got, unlike systems up north, we've got a fair period between the time you either harvest your soybean or if it gets flooded and it becomes a green manure, between then and when we plant cane. Our winters aren't particularly cold, so you've still got some warmth there to break down residues, and quite often we get some rainfall. So we thought it's worth checking whether or not all that nitrogen stays in the soil or whether or not there's actually some losses of nitrogen over winter. So what we did a couple of seasons back was we quantified nitrogen fixation in a bunch of soybean crops around the place. And then in 3 fields, we used an isotope. So my technician put an isotope of nitrogen, which is something that we can just use to track nitrogen in the field. We labelled up some soybean plants with nitrogen isotope. We then took them to the field and put them in 3 fields to see what happened to the nitrogen from the soybeans over the winter period. And if you look at that, if you look at the table that I've got in the left-hand corner over here, we have 3 fields, field 3, field 5, field 9. Fields 5 and 9 were from Korakai. So if you come over to the rainfall here, you can see this is where we put, the red part is where we put the soybeans out, and at the end of the red rainfall area is where we measured how much of the nitrogen from those beans was left in the soil. So there was more rainfall over here at field 9, which is the Ballina site, as you'd expect. So across here, this is the total recovery of the nitrogen in spring when we checked. So what we did, so all the soybean residue decomposed, we just put it on the surface. We didn't incorporate, we just put it on the surface. It all decomposed, and so we didn't have any residue left after the 5 months. And we cored soil cores down to about 900 millimetre depth, so almost a metre. And we looked at how much of our nitrogen from the soybeans was in each of those layers. I added them all up, and the red numbers here are how much as a percent of what we put on was left. So in field 3 and field 5, which was the Korakai, we got 61% and 68% remaining. So we lost somewhere between 30 and 40% at that site, or it was unaccounted for. I couldn't find it. So it might have gone up into the air, or it might have gone down and we lost it as leaching. We're not sure. Most of that, if you look in the 0 to 100, so 50% of what we found was still in the 0 to 10 centimetres. So it was right in the topsoil. If we come over to the Ballina site, field 9, where we had a lot more rainfall, we only recovered 55% of what we put on. So we lost 45% of our nitrogen over that winter fallow. Even more interesting, only 21% of that was in the top 10 centimetres of the soil. Another 20% was already down below 30 centimetres. So we presume that our losses here really are from leaching. So the heavy rainfall's pushing it, and eventually it gets below the root zone. Anywhere from sort of down to 1 metre, we presume that the cane will probably be able to get. Once it goes beyond that, we're not sure, and we consider that a loss. And so one thing to note is that I used, that was a wet season. There are a few crops that got ploughed in or were brown manured. So the residues we added were green manure soybean. So it had a much lower carbon to nitrogen ratio as you would have if you harvested your beans. You have a higher carbon to nitrogen ratio left in your residue in the straw, and that probably breaks down a bit slower. So but when you've got a green manure with a lot of nitrogen, it probably breaks down quicker. So that's one thing to note. If you're harvesting your beans, you may get slightly slower breakdown, and the results might quite not be accurate. But nonetheless, it pretty much shows that in our winters that are, you know, they're not freezing cold, we get some rainfall, there's a chance we might actually be losing some of this nitrogen over winter. So one thing we wanted to check out was whether we could grow a cover crop over the winter period and whether this might suck up some of the nitrogen. Unfortunately, measuring nitrogen using an isotope is very expensive, and I didn't have money to actually measure the nitrate losses. What we did measure in our winter cover crops, or one of the other points that we wanted to focus on, was our soil health. So what we looked at, the main targets were nematodes and arbuscular mycorrhizal fungi, or AMF. And I know Graham Sterling's been running workshops with a few others on soil health, and hopefully you've all heard of mycorrhizal fungi and presumably nematodes. So the photo there actually shows a cover crop trial with a lot of summer cover crops. That's up at Ingham that we're running. But you can sort of see what they look like. You can test a range of species in relatively large plots compared to the fallow on the left, which is your control. So down here, we're mainly looking at winter cover crops, you know, anywhere from about this time through to September, although we probably want to terminate them, take them out before September, so you've got time to prepare the land for cane and hopefully to store a bit of moisture. So the key types of cover crops you can try are cereals, and you know, people grow them. There's the odd paddock around here for grain anyway. Other legumes, so faba bean, chickpea, vetch, lentils, field peas. Then there's brassicas, which brassicas are known to biofumigate the soil. So they put out compounds through their roots. They also have them in their leaves called glucosinolates. And these things tend to be pretty toxic to a lot of fungi and to a lot of bacteria. And this is why, for example, out west, a wheat canola rotation is so effective in minimising diseases of wheat. By having a canola break crop, which is a brassica, you can effectively biofumigate or minimise a lot of wheat diseases. So that's one we're particularly keen on. The other option is you can combine all these or throw a bit of a shotgun mix in and have a mixed cover crop. And so we ran a few field trials over a couple of seasons to check them out. So I'll run you through both the trials we've run, one in 2018, one in 2019. Very different results. So this one was in Coracai in 2018 in the winter. It was a replicated trial. You can see from the shot on the right, it was set out with replicated large blocks. The yellow flowering one is mustard, and it's just hit flowering there. So we put it in late May in 2018 after soybean had been harvested a month earlier. So we were probably even a bit slow getting it in. There was a 1 month period there that we probably could have got growth, but we didn't. So we put it in a month after harvest. We threw in oats, faba bean and mustard. The sowing rates are probably quite high because we weren't sure how they'd go. And it also should be noted, we used a bean planter, and the mustard, that's a very heavy seeding rate, but it was pretty hard. It's got tiny seeds and it was pretty hard to control. So it was going everywhere. So we ended up with a very thick seeding rate. We planted 3 rows per bed, and we had three beds in each plot, and they were 80 metres long. So you can see there on the left, we have, this is the oats here, just starting to to get a bit yellow and look like a little bit short of nitrogen. And same as the mustard. This is the mustard in flower in July or August, and the lower leaves are starting to get nitrogen deficiency. So we added no extra nitrogen to this trial. We didn't give it any fertilizer. So you can see by the fact that it's already managed to grow a fair bit, it's mopping up nitrogen, but it's actually now starting to slowly run into deficiency. That photo was taken just before it was terminated, so we didn't really care about any deficiencies. We just wanted to get some carbon into the soil and potentially mop up the nitrogen. As I said, I didn't actually measure nitrogen because that's quite expensive, but the fact that they're green and they've taken it up suggests that we are mopping up some of it. So again, sown in May or late May, we terminated on the 14th of August. So we've got about 10 weeks of growth. So that's 10 weeks of growth for you. And then after the 14th of August, it was slashed down or sprayed out, slashed down, and then cane was sown on the 21st of September. So here's what happened. We got about 1.5 tonnes of biomass from the mustard crop and about 650 kilograms per hectare. These aren't huge amounts. 650 from the oats and the faba beans. One thing, that's the shoots only. So you've also got the roots putting a lot of carbon into soil, and something like faba beans has a root to shoot ratio of about 0.8. So for every tonne that you're putting above ground, there's probably about 800 kilos going below ground as well. So they are putting carbon into the soil. As I said, we didn't mention nitrogen leaching. We just looked at biomass production and nematodes and mycorrhizal fungi. So if you look over here at the rainfall graph, this is where we sowed our cover crop. Late May, we got some good rain after it in this period. So the cover crops grew pretty well, started to dry out. This arrow here is where we terminated it. And then we were quite fortunate in that after we took them out, we got a bit more rain in this period here to wet up the profile again before the cane was sown. So all in all, that was a pretty successful trial in that we didn't appear to lose any cane yield. So we measured, we didn't actually get yields off it. We just measured height at some point, and they were all the same. And visually, you couldn't see a difference. And Jeff Pye could probably talk more to it, but it didn't appear. We certainly had no negative effect from the cover crops. We didn't measure soil moisture either prior to sowing because we got that rain and presumably at least down to a reasonable depth, the whole profile was re-wet up. So what did we measure? We looked at nematodes. So this is once the cane was growing, we waited 10 weeks. We came out and took samples of the soil and the roots of the cane plants in each of the treatments. So you've got faba bean is the blue bars, your fallow, which is our control, is the orange, mustard and oats, yellow. So the two main nematodes, from what I'm told, are the two most destructive ones are the root lesion nematode and the root knot nematode. Up north, root knot nematode is a bit of a problem. Here we had none. We didn't detect any. We detected a bit of root lesion nematode, but they're very low counts. They're sort of only below 100 mostly, and that's quite low. And again, these are the error bars. So they shows a fair bit of variation between the replicates of each treatment. So really there's no difference here. So the cover crops had no effect. Same over here. It's possible that the fallow had slightly higher numbers of these spiral nematodes, but again, there's big error bars, probably not much to write home about there. Not much stubby nematode, not much stunt nematode. And these are the free living ones. They're the ones that SRA tell us these are the good guys. So notably in the free living nematodes, here's the fallow down here. There was a bit of a trend towards slightly higher nematodes or free living ones in some of the cover crops. But again, there's large errors and there's probably not too much you can make from that. Over here to the mycorrhizae. These are on the bottom axis are different types of mycorrhizae. So we used to just look in the roots under a microscope and measure the percent, what percent of roots are colonized by them. But these days you can actually use DNA methods and not only look at how much mycorrhizae are there, but what types or what families of mycorrhizae. So one of my students, Anders Claessens, does this. And as you can see here, there's 6 types that they assayed. The main one was this AMF A2. Not really sure what exactly that means. It's just a type of mycorrhizae. But again, in the faba bean plots, and he's only finished with 2 of them, there was much higher mycorrhizae in the faba bean than in the fallow. Down here, slightly more in the fallow for this little guy, but again, some large error bars. So it does appear that the cover crops might have an effect on mycorrhizae, but they didn't in this trial have much effect on nematodes. Onwards to our second trial. This one was done last year near Woodburn, established similar time as the last one, very early June, replicator block design. This one, though, we threw in some mixed species treatment. So we had a mix of oats, vetch, mustard, and lupins. And you can see here in the photo on the left-hand side, there's the lupin was just starting to flower before termination. Here's the mixed one. You've got the mustard in there, which is the flowering, cropping yellow. You've got some vetch hiding in there, oats. Over onto this side, this is the oats and the vetch. So there's your vetch, there's your oats. So once again, no fertiliser added, and they certainly have no problem growing anyway without fertiliser. So there's obviously, following soybeans, there's enough nitrogen in the soil to keep these guys going. This one we grew for a bit longer. It was terminated in early September, so it had 12 weeks of growth. So we had 3 months of growth. Cane planted in early October, and then in the trial area at least, cane replanted in early November because the cane didn't come up. And here's the story. So while in the first trial in 2018 we had no difference on cane yields, at this site it was very different. And the graph here will tell you why. This is the rainfall graph. So here's prior to planting the cover crop here. It's kind of like a graph of David Warner's batting average, this part being the Australian summer and the rest of the year representing when he goes anywhere overseas and can't score a run. So basically, from when we planted the cover crop here, all the way through to when we terminated it, to when the cane was planted, there wasn't a drop. And that explains much of our results. So we still managed to grow a fair bit of biomass. We were getting upwards of 2 tonne of carbon, or 2 tonne of dry matter being put back into the soil. Again, this is just the shoots, not even the roots. So we're definitely putting carbon back into the soil to stimulate biology, but at the expense of soil moisture. And so given that it was such a dry year, it's probably tricky to see here. This area here in the foreground is where we had the cover crops, and you can see these dark areas. That's some of the mulch after Marnie's ploughed it in. What's trickier to see, up the back here, there is a green tinge. That green tinge is all the cane emerging outside of our trial area, while in the trial area, the cane didn't even come up. So in other words, we've dried the soil out to the extent that the cane couldn't come up. One thing to note, we had fallow plots even within our trial, but even in those fallow plots, the cane didn't come up. What that tells us is we had 3 beds wide as a fallow plot. What we think has happened is the water from those plots with the cover crops in the beds next door, water's actually been drawn out of those plots sideways to be picked up by the cover crop. So that actually the entire trial area was dry enough that nothing came up. This is the resown cane a month later. And as you can see, still struggling, but by now, at least they survived, and it's all actually looking pretty good at the moment. So once again, we looked at nematodes and on mycorrhizal fungi. In this instance, we had the opposite of last time. Not much happened with the mycorrhizae over here, except, so in other words, there's the fallow. Again, huge error bars. So there's a lot of field variability in our mycorrhizal measurements. Fallow, mixed species, and this is the oats and vetch. We got a big reduction by just growing lupin. But as it turns out, lupin is a known non-host for mycorrhizae, which would explain why it reduced their numbers. So at least that we can explain. So, but when lupin was included in the mixed species, it didn't have any effect. If we come over to the nematodes though, these are our 2 bad guys again. The root knot, once again, we don't seem to find it here, at least in these 2 trials. Whereas, like I say, in North Queensland, that's a big pest. Root lesion nematode. Here we go. The fallow had higher counts. Over 1,500 was the mean, and we did get substantial, and they'll be statistically significant reductions in lesion nematode from all three of the cover crop treatments. That's the oats, here's your lupin, and here's your mixed cover crop. So in contrast to the 2018 trial where there was no impact on nematodes, even though there was very low nematode numbers, here we do get a reduction. But again, not much else happening. No root knot, nothing happening with these other three pathogenic nematodes, and in the free living, reasonable number of them, but again, big error bars, so it's pretty hard to make anything of it. So how do we, what do we summarise out of all this? We know that we can add carbon to soils. So there's one good thing about cover crops, even if you can't measure anything specific, we know that adding carbon to soils is good. And it's not just sort of residue carbon, it's fresh, juicy carbon with decent nitrogen to carbon ratios, and no doubt that's going to stimulate some biology. So there's one plus. At one site, we had no reduction in cane yields. At another site, we completely blew the cane yields because they didn't come up. That said, they were resown and are doing fine now. So season obviously has a very big impact. The mycorrhizal fungi, again, we had different effects in different sites. At one site, we had no real positive effect that we could tell, but a reduction with the lupin in lupins. And back at the Coracai site, we actually had more mycorrhizal fungi in the favour treatment than the fallow. The reverse happened with nematodes. So at one site, we had not much happen. And then at last year's trial, we actually had a reduction in the pathogenic nematodes where we grew a cover crop. We didn't measure soil nitrogen leaching, so I can't put a value on nitrogen savings. But one thing I can tell you is at the trial last year, I can guarantee you, even without measurement, there was no nitrogen leaching because it didn't rain a drop. Given that, it's very hard, even with those nematode numbers, it's hard to put a number on it. I'm not really sure, and Seb might be able to talk more to this, but we don't really know what damage is being done by nematodes. So it's very hard to know what value do you put on reducing nematode numbers. Same with mycorrhizal fungi. We know that they are, or they're supposed to be, beneficial for plants. In a lot of the regenerative farming systems, people are targeting mycorrhizal fungi, trying to build up numbers. Putting a number on this is somewhat difficult. So cover crops do seem to have an impact on nematodes, at least in some seasons. They can also affect mycorrhizae. What's the value of this? We don't know. What we do know is in very dry seasons, and once again, last year was like one of the worst seasons on record, we can impact our cane sowing. So it's very difficult to come up with a conclusion to say whether these cover crops are of net benefit or not. So what I'd like to do is continue trialling them over the next few seasons and possibly try taking them out a bit earlier. So as I said here, we grew them for 10 or 12 weeks. What happens if I grow them for a bit less? Can we still get some soil benefits and reduce the chance of using up too much moisture? So the two questions that I've put out here, and some people might have a better knowledge of this than I. What are the yield penalties associated with nematodes, and does mycorrhizal fungi have a benefit in our systems? If we know these things, then we can put some kind of value on having a cover crop. And my other question, can we grow cover crops for a shorter period, still have some benefits? And I'll leave it there.
Thank you, Terry. Any questions or comments from anyone?
Yeah, Robert Quirk, Seb, through Terry, a couple of things. A student did a master's degree here on research on losses of nitrogen. Once it gets to the oxidation front at about 60 centimetres, all the nitrogen is consumed to drive the pyrite oxidation. So, yeah, that's another factor you've probably got to bring in to think about. Mm-hmm. The other one is, look, we had no success at all with I planted ryegrass to sop up the nitrogen. All those fields that I had that in, I had to plough out, and we had Lukas ran a similar trial to yours, probably in conjunction with you. I'm not sure. Mm-hmm. and we'll have to plough that out this year. But the fallow blocks, the fallow blocks, the cane is 100%, and the others are where we had the winter crops. They just took so much water out that they nothing grew at all.
Yep. So that's pretty consistent with what we found last year. So I guess the question is are those types of seasons the norm, and are they going to continue? And if so, I think we're in a spot of bother, Robert.
Yeah. Terry, I've spoken to some guys from the Burdick, and they can get away with it because they've got irrigation. We don't.
That's right. And even beyond that, even up further where that first field trial was at Ingham, it's not irrigated, but they get, we had 2 metres of rainfall on that cover crop. Whereas here, like I say, one season we got away with it, but, if we don't get winter rainfall, or not even winter rainfall, if we don't get some early spring rainfall to refill the profile, I think it was pretty clear, and from your experience, what the risks are.
Terry and Robert, Lucas here. What if we had a better prediction about the rainfall in spring or early summer? If we had, I guess, improved climate forecasting, that could well decide whether or not either to grow a winter cover crop or when to terminate. And I think Andrew Dougal also made the point, and this is something that we have considered, Terry, previously, is terminating the winter cover crops depending on a threshold for the soil moisture content.
Lucas, there's a forewarned is forearmed group, and I'm part of that. They're working on those long-term predictions for winter, and for this year they're saying above average rainfall for winter. So I've actually planted lupins for the winter. I'll just keep an eye on them. Our local guys come out and we have a look if the soil moisture is going. We just hoe them out no matter when. So don't wait for the rain. If it's starting to dry out too much, Terry, just hoe them out.
Yeah. So I think that's, as I said there, I think that's the key is what we need to start looking at is whether or not. How long do you need to grow them for to get a benefit? Because if you put them in for a month and then you think, okay, we'll take them out, you've got a cost of sowing them, and we don't know whether you get any benefit. So it'd be interesting to know whether you actually do get any benefit if you're putting them in for a really short term. If you are, bonus. If you're not, once again, you have to question, why am I bothering? So I think I agree, Rob. I think that's what we need to do is look at different times of taking them out, and like you say, make a judgment call at some point, keep an eye on your soil moisture. And if there's no rain on the horizon, because that's, if you're pretty confident you're going to get a big dump at some point, it doesn't matter how much you use, we'll fill the profile. But unless you know that, then you don't want to use too much.
Yeah.
Okay.
I've got a question for Terry.
Mm-hmm.
Far away.
Is it possible to go back into your cane crops, Terry, and get a yield from the plots where you did get some control of your nematodes and your VAM? And the second one is, how does the nematode numbers in the two trial areas you had compare with the survey that Seb did across the 3 mill areas?
Seb, you might have a better idea.
The positive results that Terry got, I'm actually looking at our results, and it's quite correlated. And also looking at the results, I don't know if you remember, Alan and others, that we had a similar strip trial at the Dangaruba farm, and basically the multi-species strips had a significant reduction in root lesion nematodes and total plant parasitic nematodes in relation to other fallow cropping. So that seems to be similar to what Terry found. And in terms of root lesion nematodes before and after the fallow crop, the trend was like very much downward and quite significant.
Yep.
Yeah, so as to whether or not we get a yield benefit by having an impact on the mycorrhizal fungi or the nematodes, that's what we haven't really been able to quantify. Even at Jeff's place, where we were pretty sure there was certainly no reduction in yields from our cover crops, we couldn't quite quantify a benefit either. So that's what I mean. We find it hard to put a number or to put a value on nematodes and mycorrhizal fungi, other than, there's definitely value. If you're dumping a 1.5 tons of carbon or a biomass into your soils, no doubt that's going to help your soil health in some way. But putting a value on it and weighing that up against the risk of using moisture, that's where at the moment we struggle. We just don't have numbers to put on it, if that makes sense.
For what it's worth, we had an attempt at answering that question last year by setting up.
Seb, can I just interrupt you there, please?
Yeah.
Seb, your microphone isn't coming out very clear. Could you just maybe move a bit closer to it?
Okay, here I am.
Thank you.
Is it better?
Yep.
Yes, thank you.
Yes, that's better.
We started a trial at Condor last year, a pod trial, to basically try to answer that yield question in relation to nematodes. But as for Terry and Lukas, last year being quite extreme in terms of weather, so far the results haven't been very promising. So whether we used nematicide or not to basically assess the difference in nematode numbers and ultimately yield, so far it's been a negative, and the assumption is that because the weather has been very much against us, the numbers remained low to start with. So we've agreed that we defer the yield analysis until next year and reapplying the treatments again to the plots. But there is a quote based on several research projects from Graham Sterling that has been saying for quite some time that nematode might represent a financial impact to the industry, to the Australian industry of around $80 million. So I think in the current context, it's significant.
Just Alan Munro again, just a comment. My understanding from work done with soybeans and the effect on nematode species is you do get a beneficial change in the species there to more favourable nematodes being present at the end of the soybean phase. But after planting cane back into that fallow ground following beans, there's a rapid increase in bad nematode numbers again. So I'm wondering if you're chasing your tail trying to control the nematodes with cover crops anyway that do reduce the numbers in the short term, but maybe not so in the medium to long term.
Yeah, that'd be an interesting one to check too, because I tend to think you're probably right. Even if in your plant cane you get a reduction, you've still got a number of ratoons to go, and you've got to think you're growing cane year, they're going to build up. It'd be interesting to track over time and just see, but I suspect you might be right.
Yes, I'm just going back to the work that Graham Sterling superimposed over some of our compost trials, where we got that benefit of a change in nematode species from the compost applications, but I think it was pretty short-lived as well, the same as the soybean result.
Mm. Yep.
Seb, question to Lukas. We know that from some work that Tom Denmead did years ago on aerial fertilising of sugarcane, and there was very little done here this year in the Tweed, but in a normal year there's probably 1,000 hectares probably aerial fertilised, or a fair bit anyway. The losses can be up to 60%. Is there some type of fertiliser that would be better used or is worth some trial work to see if we can reduce those losses?
For aerial application of fertiliser, look, I think again the role of the polymer coated urea is to keep the fertiliser in the soil, you know, past all those lost pathways and actually supply it when the crop needs it. The issue, I guess, then comes into the 2-year cane. You know, how much nitrogen is needed in that second year and when is it needed? And I don't think any polymer coated fertiliser will actually last that long. But that's perhaps where, you know, again, you know, some of the work that Terry's doing around building carbon in soil and increasing that potentially mineralisable nitrogen. If we can increase the supply of PMN at 300 days or 400 days, that could be a way to supply that nitrogen. As far as aerial application goes, I guess how the fertiliser is applied probably. As you know, again, broadcast, I guess you've got a potential for greater loss pathways when it's banded, as Terry mentioned. I think there's that sort of inherent protection of the fertiliser band as well. So again, it wouldn't surprise me that when it's broadcast and, you know, is sitting on the soil surface, perhaps there is a greater loss pathway for the fertiliser, and maybe the polymer coat would hang on to the fertiliser longer when it is broadcast aerially, but couldn't give you a definitive answer there, Rob. Sorry.
I'll just comment once. So Rob, yeah, there's fertiliser products out there with a urease inhibitor that are designed for this. So basically their aim is to minimise the risk of you throwing urea out and not getting a shower of rain to wash it in. And I'm sure you've probably come across these products. I don't know what the price difference is. We've done a lot of work with nitrification inhibitors, not so much in cane, but certainly in rice and other crops. We haven't done so much work with these urease inhibitor fertilisers. So basically the aim is, just to give it, I'll try and be brief, you put your urea on. Usually what happens if it's in the soil or if it's wet, your urea turns into ammonium. The ammonium's fine. It'll stick to your soil and won't leach, but it can lead to losses of ammonia up through the atmosphere. But usually in our soil, quite rapidly within another couple of days when it's warm and wet, it turns to nitrate. So the aim of these urease inhibitors is it doesn't let it turn into the ammonium. So the risk that you're talking about and those losses, Rob, they're from loss of nitrogen as a gas, as ammonia. They're your losses when you top dress and don't get rain. And so this product tries to just hold it as urea for a couple of weeks, and the hope is that at some point you get a shower of rain at that time, and then the urea moves into the soil, and then it can turn into ammonia. It's funny that we might have to do some work on that. If people are actually top dressing cane like that, then it's probably worthwhile doing the work, because the Macca guys have asked the same question. They've been using it because they think they're going to get a storm. They go and throw urea out. The storm doesn't come. They want to know if they've just blown their dough. Some of them are applying fertilisers with the urease inhibitors. They want to know, again, are they blowing their dough because it costs extra, or is this stuff actually hanging around longer? So if there really is, if people are throwing out urea as a top dress at some point, I think we could probably look at some of those products if you wanted, Robert, and see whether they're actually, how long are they holding it as urea for? In other words, if they're clearly not going to, if it doesn't rain for a month or two, they're probably not going to help. If it's only a week or two, maybe they do.
Alan Munro again, just with a comment on that, Terry and Robert. We routinely use green urea if we're flying urea onto one-year-old crops as a top dressing. But also going back to work that Tropical Crops and Pastures did in New South Wales in the early 90s, where they were looking at the benefit of top dressing, supplementing your nitrogen applications on year-old cane when it's stood over to 2-year-old. And they found no benefit in that trial period from top dressing at year-old. And they got a similar result to Lukas in that the higher the nitrogen applied, the higher the yield.
So Alan, you're pretty confident when you fly on green urea, you're pretty confident that you get an effect, like it lasts longer, that it doesn't disappear?
I would be fairly confident of that. And also it's not, the extra cost is not that great. It might be $50 a tonne on top of $600 a tonne. And usually you are in a dry period, Terry, when you're applying it anyway. So it's it helps with the risk, I think.
Yeah. Yep. We might do some work anyway. I'm pretty sure the macadamia guys want us to do it and just try and work out how long you get that protection for. Anyway, it might give us more confidence to either use it or not. But.
Well, I think one of the benefits with urea in cane as a year-old dressing is you can get some leaf uptake anyway from the gas as it's volatilized. So it's not all lost even if it stays dry.
That one I don't know about, Alan. I'd have to pass on that. I don't know.
Well, unless there's any more comments or questions, we've run over 10 minutes. I think we should probably leave it there unless someone to stay beyond for the meeting or webinar will be recorded or has been recorded and will link on both Sunshine Sugar and SRA websites. Sam, am I forgetting anything? I'll just say thank you to all of you, Terry and Lukas, for presenting.
Yeah.
Sam?
Thanks, Seb. Thanks, everyone, for attending today. We hope that you found it of value. A big thank you to our presenters. As Seb said, there will be a recording available on our website later today. So thanks, everyone, for coming along, and we'll talk to you all soon.
Thanks, Sam. Thanks, Seb.
Thanks, Sam. Thanks, Seb.
Thanks, guys. And to Terry and Lukas, I'm sure you will be invited to the New South Wales Farming Systems meeting when we can all get back in face-to-face groups.
Thanks, Alan. And as I said, this data is much nicer to present when we can actually discuss each slide with a bit more animation rather than just as a webinar. So very much looking forward to presenting it in a bit more detail with you guys.
And one other comment, Alan, anytime, either now or in those meetings, if there's any other burning questions that you've got or other areas of research that you think we should look at, please sing out, because, quite often, even without funding, if it's just something simple we can take a look at, we can do it. So if anyone's got any burning questions they want us to look at, just let us know and we'll see what we can do.
Yeah, you know where we are.
Yeah, I do. Thanks very much.
Righto. See you guys.
Thanks, everyone.
Thanks, guys.
Thank you.